Hi there. In this video we will talk about the molecules which make up cells. Cells are actually made up of thousands of different molecules, but there are four types of large biopolymers made up of repeating units that make up most of the dry mass of a cell and perform most of the important functions. We'll talk about these one by one. Three of them in this video: the biopolymers polysaccharides, which are made up of sugars, membranes, which are made up of fats, and nucleic acids, both RNA and DNA. In the next several videos, we'll talk about the fourth biopolymer, proteins, which are made up from amino acids. But before I talk about the polysaccharides, I want to say one important thing about the composition of a cell. And that is true whether you're talking about bacteria or you and me, is that we are all made up primarily of water. Water is approximately 70% of each cell. The dry mass of a cell is approximately 30%. Now, of that 30%, half of it, approximately 15%, is made up of proteins. Nucleic acids, both DNA and RNA, at 7% are the next largest group. Phospholipids and polysaccharides each add about 2%. Of course, these are average values, and different cell types will deviate. So these four biopolymers make up about 85% of a cell's dry mass. And it's these four that we will talk about mostly in the first third of this course. We'll start by talking about polysaccharides, which are made up of sugars. Here we see four different views of the same sugar molecule. This is a molecule of glucose. Glucose is made of 6 carbons, 1, 2, 3, 4, 5, and a 6th carbon above the ring. It's a 6-membered ring because we have one oxygen atom in there. So in the ring there are 5 carbons and 1 oxygen. This is referred to as a pyranose ring. The stereochemistry at each of these carbons is very important, whether the hydroxyl group is below or, in this case, above the ring. Different orientations will actually be different sugars, and they will have different functions inside the cell. This is an example here showing the stereochemistry. This is the chair conformation. You might remember that that's more stable than the boat conformation for glucose. And here is a ball and chain model, and here is a space-filling model. This is the most accurate representation of the molecule. However, for most of us, it's not as clear in depicting what the structure is. For instance, you might be hard-pressed to see that this is a 6-membered ring here. The number of carbons in a sugar helps define our nomenclature for it. Here we see a triose made up of 3 carbons versus a hexose with 1, 2, 3, 4, 5, 6 carbon atoms. Also, these sugars are referred to as aldoses because they have an aldehyde group at the end of them. But we're only showing here the linear conformation. These sugar molecules can isomerize between linear conformations and ring conformations, as we see here for the 6-carbon glucose sugar and the 5-carbon ribose sugar. The way these sugars cyclize is that the hydroxyl group on the 5th carbon will perform a nucleophilic attack on the carbon that has the aldehyde. The aldehyde oxygen will be converted to a hydroxyl group, and the 6th carbon will be above the ring here. So in the end, you have a 6-membered ring with 5 carbon atoms and 1 oxygen atom. This is the sugar ribose, which has 5 carbons. The cyclization mechanism is similar. The hydroxyl group of the 4th carbon attacks here at the carbonyl group, which will make a 5-membered ring. Ribose is the sugar of RNA. If this hydroxyl group were changed to a hydrogen, then ribose would become deoxyribose, and we would have the sugar for DNA. On the right side of the figure, we have a graphic that highlights the stereochemical aspects of the hydroxyl groups. If we look at the difference between galactose and glucose, we see that it's only the orientation of the hydroxyl group at this 4th carbon position, and yet the two sugars will be used quite differently. Now, polysaccharides are complex chains of sugar. At the bottom, we show a linear chain, but they can also be branched. We have 4 different glucose molecules connected. This is a starch called amylose. The individual sugars are connected from carbon 1 to carbon 4 by a glycosidic bond. In this case, it's called an alpha linkage because the oxygen atom of the glycosidic bond is below the ring structure. Now we'll talk about fats and lipid membranes. Fats are made up of fatty acids, which have long hydrocarbon tails, just carbon and hydrogen. They're called fatty acids when they have a carboxylic acid group here as the head group at the end. These are extremely hydrophobic overall and will avoid water at all costs. The fatty acids can be comprised of different types. One type shown on the right here is a fully saturated fatty acid. That means they're all single bonds, no double bonds, and it is referred to as saturated because it has as much hydrogen attached to the carbons as possible. Now when we talk about unsaturated fats, that means there's a double bond somewhere in this chain. If both connected carbons are on the same side of the double bond, we would refer to this as a cis fatty acid. And if it were a trans fatty acid, the carbons would be on opposite side and the tail would come down somewhat in this direction. As you know, there are health concerns with trans fatty acids, and certain municipalities are trying to ban them now. When we look at the space-filling models, we notice that there's a real large consequence from the double bond, a geometric consequence that affects the overall shape of the chain. This unsaturation makes the fatty acid highly kinked. Now imagine if you're trying to pack many fatty acids together, as closely as they can be packed. You can imagine that you can pack the saturated fats much more closely and easily than the unsaturated fats because of the kinks. So fats that contain unsaturated fats will be less densely packed. They'll be more liquidy, like for instance olive oil, whereas saturated fats would be more solid. That's the main difference between a saturated fat like butter, which is solid at room temperature, and olive oil, which contains the unsaturated fats and is liquid at room temperature. Now, in addition to fatty acids, when we're building up biopolymers of fats, the molecule glycerol, a 3-carbon molecule with 3 hydroxyl groups, can be included. As depicted here, glycerol can react with 3 fatty acid chains to make a triacyl glycerol, also referred to as a triglyceride. You'll notice that each of these fatty acids need not be the same. In this example, we have 2 saturated and 1 unsaturated chains. You can imagine that combinatorically, there are many different types of triacyl and diacyl glycerols in a cell. This is the depiction of a phospholipid. It's similar to the triglycerides we just saw, with being held together by a molecule of glycerol shown here in green. The major difference is that one of the hydrophobic fatty acid chains has been replaced with a hydrophilic head group. These amphipathic molecules, containing both hydrophobic and hydrophilic sides, are the main constituents of membranes. In this case, the 3rd hydroxyl group is connected to a phosphate group, and then the molecule referred to as choline, which is similar to amino acids. So this is the molecule phosphatidylcholine. These molecules are one of the primary constituents of eukaryotic membranes. Again, this is the region below the glycerol backbone is going to be very hydrophobic, and this is going to be in the center of the membranes. This portion, the hydrophilic region, will be facing outwards into the aqueous environment. We also see on this slide how the individual phospholipids are organized to form a membrane. First, you'll notice that there is a bilayer. For each leaflet of the bilayer, the hydrophilic heads are oriented outwards, facing the water. The hydrophobic tails of the 2 leaflets are pointed inwards and interact with each other. They are sequestered away from the water. Now this is a cutaway view. We do have some problems still. The sides of these lipids are exposed, and that cannot exist. We'll see in future lectures about how the cells are going to solve that problem. We'll now talk a little bit about nucleic acids. Nucleotides are the subunits of DNA and RNA, and there are three major parts to a nucleotide. There are phosphate groups, there's the sugar group, and together these two groups will form the double helical backbone. Note, and we'll talk about this a lot, and you will forget about it many times, the phosphate group is negatively charged. That means the backbones of DNA are going to have lots of negative charges. DNA is a polyanion. And the third part of the nucleotide is the base, the nitrogenous base, which is what it's called. They can vary. For DNA, these bases will be A, C, T, or G. For RNA, U will replace T. Now there are two main types of nitrogenous bases that are used in nucleic acids, pyrimidines and purines. In DNA, we have adenine, guanine, thymine, and cytosine. And in RNA, instead of thymine, we will have uracil. But uracil and thymine code for the same information content. Each time you have a base pair in a nucleic acid, you will have one pyrimidine and one purine. The way I remember which is that the longer word pyrimidine refers to the smaller one-ring structure, and the smaller word purine refers to the larger two-ring heterocyclic structure. The second part of nucleic acids is the phosphate group. In the final structure of DNA and RNA, we have a single phosphate unit. But the original building blocks, the nucleotides, are triphosphates, so they have 4 negative charges. We will lose 2 of these 3 phosphate groups as DNA is being synthesized. The third constituent of nucleic acids is the sugar pentose. It's either ribose or deoxyribose. It's a 5-carbon sugar, 1, 2, 3, 4, and 5. Used in RNA, we have this hydroxyl group at the second position, the 2' position. And in deoxyribonucleic acid, in DNA, we've lost the oxy of the hydroxyl group, so it's deoxy, and we've replaced it with a hydrogen. So this is a small molecular change, but it has large consequences for function and structure of these molecules. Kind of amazing when you think about the small change having such a profound effect. Now you might wonder why these atom positions are called primes: 1', 2', 3', 4', and 5'. That's only to indicate positions on the sugar. The positions of the nitrogenous base would be without the primes. So we're going to talk a lot about the hydroxyl group on the 5' carbon and the hydroxyl group on the 3' carbon. The reason we will talk so much about the 5' carbon and the 3' carbon is that because the bond connecting nucleotides in DNA is between the 3' carbon and the 5' carbon. They are bonded together via a phosphate group, forming a phosphodiester bond. You always add new nucleotides of the growing nucleic acid chain to the 3' end of the nucleic acid. Although we'll focus in the first part of the course on nucleotides' role in DNA and RNA as for information storage and information utilization, many of you will remember that these molecules can perform other roles inside the cell. In particular, you'll remember that ATP is one of the main high-energy intermediates that can be used for energy storage or utilization inside the cell. As mentioned, ATP has 4 negative charges in close proximity. You know that negative charges repel each other. So for them to be in this close configuration, that's a high-energy form. When ATP is cleaved into ADP, some of that negative electrostatic repulsion will be relieved, and the energy loss will be available to do work if you can capture it. One of the ways it is captured is by inducing a conformation change in a protein. ATP can also be converted to a form called cyclic AMP, with a bridge between the 3' and the 5' hydroxyl groups. This molecule is often used as a signaling intermediate. The last point to make about DNA in this particular lecture is a point you'll see over and over again. This is already the second time you're seeing it in this lecture, and that is that DNA is always synthesized from the 5' end to the 3' end. We're always going to add a new nucleotide down here. Here you see 4 nucleotides, G, A, T, and C, and the 4 different bases you can have. And their particular order is how information is stored in the nucleic acid backbone. Alright, that includes our introduction to 3 of the biopolymers: polysaccharides, lipid membranes, and nucleic acids. Next, we'll consider proteins. Thank you for listening.